Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Welcome to the Deep Dive. We're here to take you into the heart of cutting-edge research and pull out the insights that matter to you.
And today, wow, we have a fascinating topic.
Absolutely. We're looking at something that, well, it really reshaped things recently. Messenger ribonucleic acid vaccines.
That's right. Thinking about the COVID-19 pandemic, the
Yeah.
speed and, frankly, the success of those vaccines was just remarkable.
It really was, almost felt like magic. But it wasn't magic, was it? There's some incredibly clever biology going on underneath.
Exactly. A kind of biological trick, you could say.
And that's our mission for this deep dive, isn't it? To unpack that trick.
Precisely. We want to explore the molecular mechanism, how this modified ribonucleic acid, which is the engine in these vaccines, and potentially many future drugs.
How it manages to get into our bodies and do its job without causing a massive unwanted immune overreaction.
It's like biological stealth technology. And we're looking at the very latest research, fresh off the press, that explains how it works.
Okay, so where are we getting this info?
We've got two excellent sources, both published this year, 2025. There's a great health news piece summarizing it, and then the detailed scientific paper itself.
And they both come from a research team at Ludwig Maximilian University of Munich.
That's the one. Really groundbreaking work.
Fantastic. Now, before we dive headfirst into the mechanism, maybe we should clarify some key players here, some terms.
Good idea. So we'll be talking a lot about ribonucleic acid, RNA. That's the general molecule carrying genetic instructions.
And specifically messenger RNA, or mRNA.
Exactly. mRNA carries the specific blueprints for making proteins. We also need to understand the innate immune system.
That's the body's first responders, right? The immediate reaction force.
Perfect analogy.
Yeah.
And within that system, we'll focus on specific sensors called pattern recognition receptors,
Okay.
particularly a type known as toll-like receptors. Think of them as alarm systems.
Got it. RNA, mRNA, innate immunity, toll-like receptors. Let's go.
Okay, so picture this.
Yeah.
Your body has this constant surveillance happening. That's the innate immune system.
Always scanning, looking for trouble, like viruses, bacteria, things that shouldn't be there.
Precisely. And the key sensors doing this scanning, particularly for foreign RNA, like from a virus, are these toll-like receptors. We're especially interested in toll-like receptor 7 and toll-like receptor 8.
And where are they? Just floating around?
No, they're strategically placed inside cellular compartments, little bubbles inside cells called endolysosomes.
Okay, inside the cell. So how do they actually spot the intruder RNA? Do they just see it floating by?
It's a bit more involved. For these toll-like receptors to actually sound the alarm, the RNA they're looking for usually needs to be broken down first.
Broken down.
Yeah, by specific enzymes, sort of like molecular scissors, also inside those endolysosomes. They chop the RNA into smaller fragments.
And those fragments are what the receptors recognize.
Exactly. It's the little pieces that trigger the alarm, not necessarily the whole molecule intact. That's a crucial point.
Okay, that makes sense. So the alarm system needs these broken bits.
Right. And this brings us neatly to the big discovery from the Munich team.
Led by Professor Veit Hornung and Professor Thomas Carell.
Yes, and with really key work from Marlene Baruti and Mirko Wagner. They figured out the molecular details of how modified RNA avoids this whole process.
This is the core of it then. What did they find?
So here's the breakthrough. They showed, really for the first time with this level of detail, that if you modify the RNA, specifically, if you swap out one of its normal building blocks, uridine, for a slightly different one called pseudouridine,
Okay.
that modified RNA becomes much harder for those enzymes, those molecular scissors, to cut up.
Ah, so the fragments aren't generated as easily.
Exactly. But that's not all. There's the second part to it.
Oh.
Even if some fragments are produced, the toll-like receptors themselves often fail to recognize the fragments that contain that pseudouridine.
Wow. So it's a double whammy.
Yep.
Harder to cut and the pieces are harder to see.
Precisely. A really effective two-layer defense against detection.
Now, this idea of using pseudouridine, it rings a bell. Wasn't there earlier work on this?
Absolutely.
Yeah.
This builds on really fundamental research from back in 2005.
Okay.
That study was the first to show that putting pseudouridine into synthetic mRNA dramatically dials down the immune response. That discovery really paved the way for everything that followed the RNA therapies and vaccines we have now.
And we saw this used in practice, didn't we? Like in some of the COVID-19 vaccines?
Yes, exactly.
Hmm.
Many used a related modification, something called N1-methylpseudouridine.
N1-methylpseudouridine.
Okay, and the Munich team found that this modification also largely evades recognition by the toll-like receptors. Similar principle.
So why is this evasion so critical? What does it mean for the vaccines and potentially other treatments?
Well, it's fundamentally important. This mechanism is why these modified messenger RNAs don't trigger massive inflammation.
Which would be a big problem.
A huge problem.
Wow.
If the vaccine itself caused a really strong, nonspecific inflammatory reaction, it could cause significant side effects and potentially be less effective at generating the right kind of immunity. This stealthiness is key to their safety and success.
Okay, so you mentioned this is like a two-layer system. Can we dig a bit deeper into those layers? You said it's not just about being not processed.
Right, it's more nuanced. The paper really breaks it down nicely.
Yeah.
Layer 1 is this impaired enzymatic processing.
The cutting problem.
Exactly. They identified specific enzymes, one called RNase T2 and others known as PLD exonucleases, inside those endolysosomes. Their job is to chop up RNA into those little signal molecules, the ligands, that activate toll-like receptors 7 and 8.
Okay.
But the study clearly shows these enzymes struggle with RNA that contains pseudouridine, or that N1-methylpseudouridine. They just don't process it efficiently.
Like the scissors are blunt for that specific material.
That's a great way to put it. The unique chemical structure of pseudouridine seems to make the RNA strand a poor target, maybe changes its shape slightly, making it hard for the enzymes to latch on and cut.
Fascinating. Okay, that's layer 1. What about layer 2, the receptor avoidance?
So even if some modified fragments do get produced, there's this second safety net at the receptor level. For toll-like receptor 8 or TLR8, it basically doesn't recognize pseudouridine well in its main binding spot. It sort of neglects it.
So TLR8 ignores pseudouridine fragments.
Largely, yes. Interestingly, though, they found that N1-methylpseudouridine, the one used in some vaccines, can actually activate TLR8 directly sometimes, even though it resists being cut. It's a subtle point, but potentially important.
Huh, okay. Now what about the other receptor, TLR7?
Toll-like receptor 7 also seems to largely overlook RNA fragments that contain pseudouridine, specifically in its second binding pocket, which is important for its activation.
So both layers are really working together. Reduced cutting and reduced recognition of whatever is cut.
Exactly. It provides what the paper calls a molecular basis for immune tolerance to pseudouridine-containing RNA. The structure just makes it stealthy.
It's such an elegant solution, really. How did the researchers actually pin all this down? What methods did they use?
They used some pretty sophisticated techniques. A core method was quantitative mass spectrometry.
Okay, what does that let you do?
It's incredibly sensitive. It allows you to precisely measure and identify the different RNA fragments produced after the enzymes try to cut them. You can see exactly what pieces are being made or not made and how much.
So they could literally see the difference in fragments between normal RNA and the modified RNA.
Precisely. And they didn't just test tiny bits of RNA. They used short ones, oligonucleotides, but also much longer RNA molecules, around 2,500 nucleotides long.
Which is closer to the size used in actual vaccines.
Exactly. So comparing the unmodified versus the pseudouridine or N1-methylpseudouridine versions in these more realistic contexts.
Makes sense. Did they look beyond just the molecules in a test tube?
Oh, yes, definitely. They tested the immune response in actual human immune cells, primary monocytes and plasmacytoid dendritic cells, which are key players carrying these toll-like receptors.
See how real cells react.
Right. And they even went further and did studies in mice to confirm that these mechanisms hold true in a whole living organism, an in vivo setting.
That's quite comprehensive. Molecular level, cellular level, and in a living system.
Yeah, it really strengthens the findings. It shows this isn't just an artifact of a test tube experiment.
So stepping back a bit, what are the broader implications here? This seems fundamental.
It really is. On a basic science level, it gives us huge insights into how our bodies distinguish self from non-self at the RNA level. Why doesn't our immune system constantly attack our own RNA, which also contains some natural pseudouridine?
Right. Our own cells use pseudouridine too.
They do. So understanding how these modifications contribute to tolerance helps explain this fundamental process of self-avoidance, which is obviously critical for preventing autoimmune reactions.
And thinking about the future of medicine.
Well, these findings are incredibly valuable. They provide, as researchers say, starting points for the future targeted development of RNA-based drugs.
How so?
Because now we understand the specific molecular tweaks needed to make RNA stealthy to the innate immune system. Scientists can use this knowledge to design better, safer RNA therapies for all sorts of things beyond vaccines.
Like treatments for genetic diseases or maybe even new types of cancer therapies.
Potentially, yes. Anywhere you need to deliver genetic instructions via RNA without triggering a massive, unhelpful immune flare-up. This understanding gives designers more control.
Of course, no single study answers everything. Were there limitations acknowledged? Things we still don't fully understand.
Absolutely. Science always builds step by step. One complexity is that our own cells have a whole machinery for adding pseudouridine naturally to our RNA.
Right, the pseudoridylation machinery.
Yes, and because that machinery is essential and complex, it's hard to completely isolate the exact role of just pseudoridine in self-recognition compared to maybe other RNA modifications or other factors involved. There might be multiple layers of self-recognition.
So pseudoridine is a big piece of the puzzle, but maybe not the only piece when it comes to telling our own RNA apart.
That's a fair way to put it. Also, it's really important to remember the scope of this study.
What do you mean?
It focused specifically on the innate immune response, that immediate toll-like receptor-driven alarm system.
Okay.
It wasn't designed to look at the effects of these modifications on the adaptive immune response.
Which is the part involving antibodies and T cells. The long-term memory part, crucial for vaccines.
Exactly. So how pseudoridine or RNase T2 affects the development of that long-term protective immunity from a vaccine? That's still an open question and an area for more research.
Got it. So this study brilliantly explains the stealth aspect for the initial immune encounter, but the downstream effects on adaptive immunity need further study.
Precisely. Which is exciting. It opens up new questions.
Definitely. So to kind of wrap things up, this deep dive has really peeled back the layers on this incredibly elegant molecular strategy.
Yeah, this dual mechanism resisting the enzymatic cutting and then evading the receptor recognition, it's how modified RNA achieves its immune stealth.
And that stealth is what unlocked the door for modern messenger RNA vaccines and holds so much promise for future therapies.
Absolutely. A fantastic piece of biological engineering, understood now in much greater detail.
So, as we leave you to think about this, consider this fascinating angle. Now that we have this deeper understanding of how pseudouridine acts as a molecular cloaking device, what new kinds of RNA therapies could this enable? Think beyond vaccines.
Yeah, and could we fine-tune these modifications even further? Maybe make RNA completely invisible to the immune system when needed.
Or maybe even engineer specific modifications to trigger a controlled immune response for certain diseases where that might actually be beneficial, like certain cancers.
It raises so many possibilities. What new questions does this make you wonder about regarding how our bodies tell friend from foe right down at the molecular level?
Thanks for tuning in to the Health Headliner podcast by Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.